Today we will discuss the factors that is responsible for late stage activation of macrophages, and in the conclusion what they have find out is that the CCR5 cytokines in the vicinity of CD8 plus T cells are necessary to differentiate the macrophages in the M1-type phenotype. So how they come up to this conclusion, we will understand through the results that are presented in this figure. So first what they did is they analyzed two kind of data. First one is the responder versus untreated cells, and second one is the vaccine plus bispecific antibodies treated versus untreated tumors. So in the first case what they have find out is that in all or in both the cases they have find out is that this CCL3, CCL4, CCL5 cytokines are increased along with their receptor, that is CCR5, and they have also seen that IFNNA1 and IFNNAB and LTBR are increased in the responder case, whereas LTB and IFN gamma is not shown in here, is increased in both the cases. So once they find out this analysis using the transcriptomes data, they validated their analysis in the experiment where they treat the tumor cells with the supernatant of activated T cells, and what they find out is that there is a upregulation of the CCL3, CCL4, CCL5 and IFN gamma, both in the spleen tumor and both in the spleen cells and also in the tumor cells. This indicates that the transcriptomic analysis is correct and there is an increase in the cytokines in the presence of activated T cells. And now some of the results are presented in the supplemental figures, but I will provide you a brief overview that after this they checked, they wanted to see this, they wanted to understand how these macrophages get differentiated to the M1 type, how these macrophages are monocytes differentiated towards this M1 type macrophages and less towards M2 type macrophages. So from the previous literatures they find out that this IFN gamma plays a role towards differentiating macrophages or monocytes into the M1 type macrophages. So and IFN gamma can interact with this CCR5, or this CCR5 interactions will cause CCR5 interactions will cause more formation of this IFN gamma. So what they did is they inhibit this CCR5 and check that IFN gamma gets reduced, and when the IFN gamma gets reduced, let me correct this thing. So first what they did is they inhibit this IFN gamma and check whether the monocytes differentiated towards the M1 type macrophages gets affected or not. And what they find out is that when they inhibit this IFN gamma, they see a reduction in the M1 type macrophages differentiated to the monocytes differentiated towards the M1 type macrophages. They see a decrease in those differentiation when they inhibit IFN gamma, and when they inhibit CCR5, they do not see too much difference in the macrophages. However, they see a decrease in the INOS population. That is INOS populations that are formed in the CD8 plus T cells. So this indicates that CCR5 affect the enzymes, that is INOS, in the activated CD8 plus T cells. However, this IFN gamma affects the macrophages differentiation. So combining this, what they come to a hypothesis that CD8 plus T cells should be in the vicinity of the CCR5 ligands, so that this INOS will get upregulated and it will trigger these IFN gamma formations, and that will cause the differentiation of the macrophages or monocytes towards the M1 type macrophages. In order to check that hypothesis, they did the dual staining of first they did it for CD8 plus T cells and F480. which are next to F480, and what they analyze is that they wanted to check whether CD8 plus T cells and this F480 are next to each other or not. So this is what they find out. For this is the example that they have calculated. The CD8 plus T cells is surrounded by F480 cells, and when they inhibit the CCR5, they see a decrease in this uh, arrangement. That is CCA8. That is CD8 plus T cells is less surrounded by F480. So this indicates that CCR5 will cause reductions. Inhibition of CCR5 will cause reduction in this IFN gamma, or CCR5 plays a role towards this IFN gamma cytokines, and that in turn attracts the differentiation of monocytes towards macrophages. So with this, CCR5 indirectly plays a role towards differentiation of M1-type macrophages to M1-type macrophages from monocytes. And they also check the effect on the INOS populations, and this is, we can see that the CD8 plus T cells is surrounded by the INOS, is tumors with the INOS, and when they inhibit the CCR5, they see reductions in these INOS populations, and these things are quantified. Panel C and D are quantified in the panel E and F respectively. So in the panel E, they can see that the average number of F480 cells next to CD8 plus cells are high when the CCR5 is not inhibited. However, when the CCR5 is inhibited, they see a decrease in these populations. And when they did the similar experiment on the B16F10 mice models, they see a similar results. That is, when they use the vaccine plus bispecific antibodies, they see a more population of CD8 plus cells close to F480 or vice versa, and they see a decrease in those neighboring populations when they use this CCR5 inhibitors or antibody to neutralize CCR5. And in terms of the INOS, they also see the similar results. That is, when the RMA-QA1 B model is xenograft mice model is used, they see a increased population of INOS next to CD8 plus T cells. That is, tumors with INOS is next to CD8 plus T cells. And when they use the CCR5 neutralizing antibodies, they see a decrease in those populations. And similar results they have observed for the B16F10 mice model, xenograft mice model. So from this, they come to a conclusions that the late stage activation of monocytes towards M1-type macrophages occurs by activated C cells when the CCR5 cytokines are in the vicinity of these activated C cells. So I hope you understood, and these things they have validated, that is panel C and D are validated used on the tumor microenvironment. So from this, I hope you understood how they come up to these conclusions. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.